SwayamPrabha. Digital India. Educated India.
Dear students, you are welcome in this class. I am Dr. Dinesh Kumar, Principal Scientist from Division of Agronomy, ICAR, Indian Agricultural Research Institute, Pusa Campus, New Delhi. This lecture pertains to the course Agronomy of Oilseed, Fiber and Sugar Crops, and the title of the lecture is Nutrient, Water, Weed, Insect Pest and Disease Management and Rating and Quality of Sannhemp. So overall learning objectives of this lecture are to explain the nutrient, water and weed management practices in sannhemp, means how one should go for nutrient management in this crop, what should be the options for water management and how to control the weeds. So these subjects will be covered. In addition to that, you will learn the management of insect pests and diseases and rating and fiber quality. Particularly this crop when grown for fiber purposes, then how to go for rating and how the quality of fiber is important. So all these aspects are going to be covered in this lecture. Now you can see the crop of sannhemp. This is quite green, lush green you can say, and it yields fiber. It also used as green manure crop or as a fodder crop. So there are three important uses of this crop. Now glossary, facultative parasite, an organism that can live both as a free living entity and as a parasite, that is called as facultative parasite. Grubs are actually larvae or caterpillar which do not have their legs. So immature larval stage of certain insects, particularly beetles or coleopterous insects, commonly found in soil and have C-shaped body. So white grub, many of you might be knowing. Hemicellulose, this is heterogeneous group of polysaccharides present in plant cell walls along with cellulose, pectin and lignin having branched structure, that is hemicellulose, which is part of the fibers. Leg hemoglobin, molecule similar to hemoglobin of animals, plays a vital role in symbiotic nitrogen fixation process in legume plants. So this leg hemoglobin is found in the roots of leguminous plant and it helps in the fixation of nitrogen by legumes. Phyllodes, it is a kind of disease and caused by mycoplasma kind of organism and a pathological condition in plants where floral parts, petals, sepals and reproductive units transform to leafy structure, means the flower is overall transformed into leafy structure and there will not be any pollination, fertilization or seed development. Phytoplasma, specialized bacteria that infect plant phloem tissues and transmitted by insect vectors, phyllodes of sannhemp. This is caused by phytoplasma. Pustules in plant, small raised blister-like lesions filled with spore masses. In plants, they often indicate a fungal infection where pustule contains spores of pathogens. Root nodules are specialized structure formed on roots of certain plants like legumes as a result of symbiotic relationship between with nitrogen fixing bacteria. Many of you are aware of this fact that rhizobia infect the legume roots and then they form these nodules where nitrogen gets fixed. Sedges, group of grass-like plants belonging to the family Cyperaceae. So in fact there are three kind of weeds in crop. One is broadleaf weed, other is narrowleaf weed, and third category in between these is sedges, means the plants belonging to family Cyperaceae are called as sages. Sporodochium. Sporodochium is a small compact stroma which is mass of hyphae formed on host plants parasitized by certain fungi bearing conidiophore on which conidia are formed. So, conidia are kind of spores. They born on conidiophores. Steeping, process of soaking sannhemp plants in water to extract the fiber after retting. So, steeping. Vascular bundles in plants. These bundles are composed of xylem and phloem which transport water, nutrients and sugar throughout the plant. So, under most circumstances, xylem translocates the water and mineral nutrients and phloem translocates the food material manufactured by the plant. Now, see nutrient management in sannhemp crop. So, this crop is leguminous crop and it can fix atmospheric nitrogen in their nodules. Thus, it requires a starter dose to meet nitrogen need at early stage until nodules become active. So, for most of the leguminous crops or plants, you require a starter dose because in the beginning at seedling stage, plant is not able to make or manufacture nodules where nitrogen can be fixed. So, therefore, for initial growth, you need a starter dose of nitrogen which is normally 20 kg per hectare. But it requires phosphorus and potassium at the rate of 40 kg per hectare recommended. It promotes good growth of the crop. And we need to remember that all these fertilizers, nitrogenous, phosphatic and potassic, are to be applied basally at the time of sowing. And as far as possible, phosphatic and potassic fertilizer should be placed below the seed or near the seed. Calcium and magnesium have been found to promote nodulation of sannhemp. Combined application of phosphorus and potassium, P at the rate of 20 kg per hectare and K 40 kg per hectare, produce highest fiber yield of 1.06 ton per hectare at Barakpur. Reported by Saha and co-workers 2009. Total dry matter, fiber yield and nutrient uptake increased significantly with application of potassium and sulfur each at 40 kg per hectare. So, you can see that now certain plants, certain crops, particularly this sannhemp crop is responding well to the sulfur, but this is at location specific deficiencies where plants respond to sulfur. So, many parts of our country are getting deficient mainly because of less use or decreased use of single superphosphate and sulfur containing pesticides or fungicides, and also leaching of sulfate occurs, and there may be some more reasons also. So, use of potassium and sulfur also increase number of nodules and crude protein content of leaves at Pratapgarh, Uttar Pradesh. So, again response to potassium and sulfur was achieved in Uttar Pradesh also, in West Bengal also. Application of micronutrients like iron, manganese, zinc, molybdenum, boron, these are the symbols, showed a stimulatory effect on growth and nodulation in sannhemp. Application of zinc sulfate at the rate of 20 kg per hectare and borax at the rate of 5 kg per hectare gave higher fiber yield of 0.91 to 0.98 tons per hectare at Pratapgarh, Uttar Pradesh, and recommended for the region. So, now you see new deficiencies of nutrients are coming up, and in Uttar Pradesh, Pratapgarh, application of zinc and borax is also recommended for this crop. Foliar spray of molybdenum at the rate of 50 gram per hectare improved nitrogen fixation by crop. Iron increased nitrogenase activity and production of lag hemoglobin in sannhemp crop. So, iron is important for formation of lag hemoglobin. Micronutrients required in very small amounts be applied only when deficient in the soil. So, in case of micronutrients, the best option could be to go for soil testing, and based upon soil test, micronutrients should be applied. Application of organic matter at the rate of 5 ton per hectare or organic manure is essential as it improves soil physical, chemical and biological properties, and it also supplies the nutrients after decomposition, and of course it is good for the growth of the rhizobia bacteria also. So it has been reported under certain conditions that addition of organic manures induces or increases nitrogen fixation by the legumes. Organic carbon, available nitrogen and phosphorus of soil improved with application of manure and indicating a possible increase in biological nitrogen fixation. So overall, addition of organic manure is quite advantageous. Application of potassium and sulfur individually at 40 kg per hectare each increased fiber yield by 20 to 21%. Means really definitely it was a good response to potassium and sulfur, where on an average 20% increase in fiber yield was noticed. Seed inoculation with rhizobium and PSB. PSB is phosphate solubilizing bacteria. So these bacteria solubilize the unavailable phosphorus into available phosphorus. So seed treatment with rhizobium and PSB along with 75% recommended fertilizer dose gave higher fiber yield. Means if you use these rhizobium and PSB, then you can save about 25% nutrients and you will get good fiber yield, maybe 0.76 tons per hectare. It also improved soil fertility and reduced fertilizer requirement by 25% at Pratapgarh, and hence it is recommended for this region. Now, after going for nutrient management, let us see water management aspect in sannhemp crop. So sannhemp crop in general is grown during Kharif season as a rain-fed crop. Sowing in mid-April requires 4 to 5 irrigations at 15 to 10 days interval, resulting in higher fiber yield. So in certain cases, it can be grown during the summer period. But in summer period, for example, in Pratapgarh district of Uttar Pradesh, you need to apply 4 to 5 irrigation at an interval of 15 to 20 days, and you can get good fiber yield. So therefore, under certain conditions, it can be grown as an irrigated crop also. Simultaneously, it does not tolerate waterlogging. Thus, the need-based drainage should be accomplished. Seed crop is also usually grown as a rain-fed crop. Enough soil moisture is available at sowing and no irrigation is required at early growth stages. If some pre-monsoon shower comes, moisture is there, then you need not to go for pre-sowing irrigation, crop can be sown. But 2 to 3 light irrigations are given at seed setting and pod maturation stage. If farmer is growing this crop for seed purposes and there is no rainfall at the time of flowering or pod development, then at those stages irrigation should be given. If only one irrigation is available, it should be given at pod setting and maturation stages in seed crop. Now, go to the weed management. Generally, sannhemp crop does not face much weed problem during its growth period except at the initial establishment phase. So, initial phase is critical for crop weed competition. If weeds are not controlled during this critical period, then there will be maximum loss in crop yield. Therefore, weeds should be controlled in initial stages. However, at later stages, it is dense canopy smoothes the weed growth. Mainly because of shading effect, it can suppress the weeds. Some most common weeds of sannhemp fields are given in the next slide. So you can see the major weeds of sannhemp crop: sedges, Cyprus rotundus L. Coming to grasses. So dear students, grasses are also called as narrow-leaved weeds. Their leaves are narrow, means they have more length than the width, and they are also called as monocot weeds. So it includes Cynodon dactylon, Dactyolactinium egyptium, Echinocloa colonum L, and Eragrostis tremula, Leptocloa filiformis, and Saccharum species. So these are the major grassy weeds in sannhemp crop, and broadleaf weeds are Euphorbia hitta, Eclipta alba, Commelina species, and Ipomia species. So these are broadleaf weeds or dicot weeds of this crop. See, you can identify these weeds and see this is Cyprus rotundus, this is Cynodon dactylon or dhoop grass, and then you can see Dactyloactinium egyptium, and this is Echinocloa colonum. This is a very common weed in cereal crops also during Kharif season or rainy season. Now you see Eragrostis tremula, you can see Leptocloa filiformis and Saccharum species. Now see some broad-leaved weeds, Euphorbia hitta and Eclipta alba. This is Commelina species having bluish or light bluish color of flower, and Ipomia species is also there in this crop. Now how to manage the weeds? Because we know in any crop they are going to do damage. The crop losses may be there, particularly they compete for space, they compete for nutrients, water and light, etc. And finally they reduce the growth of the crop or plants. So hence their control is necessary or their management is necessary. So what can be done for weed management? One hand weeding may be provided at 20 to 30 days after sowing, depending upon weed growth, because this is very critical period. At this period there should not be any weeds. For chemical weed control, Mosgidis and Vej 2011 reported that pre-emergence spraying of pendimethalin at the rate of 1.15 kg AI per hectare or Imejata pyre at the rate of 0.07 kg acid equivalent per hectare controlled Digitaria sanguinalis by 76 to 78% and Eragrostis species by 93 to 94%. So these are important herbicides that can be used if somebody is interested to control weeds by herbicides. Let us see some important diseases and their management in sannhemp crop. Among different diseases in sannhemp, vascular wilt, sannhemp mosaic and anthracnose are major ones, but several minor diseases like leaf spot, leaf blight, rust caused by Euromyces decoratus and twig blight caused by Coanephora cucurbitarum had also been reported in sannhemp. So there are number of diseases, but basic diseases are vascular wilt, sannhemp mosaic and anthracnose. So let us see vascular wilt. As the name suggests, the plant will start wilting. So sannhemp wilt is caused by Fusarium udum butt F species Crotalaria. Infected plants wither, leaves start yellowing and ultimately plants turn brown and die. So you can see on the right side the plants are showing yellow leaves, and these leaves are drooping. They are just hanging from the branch or hanging from the stem, and then they can dry up. The whole plant many times can dry up if there is severe attack of the wilt. So what happens? This fungus enters the xylem vessels of the plant and it stops the circulation of water and nutrients. So you can see the symptoms, more symptoms. It is drying of the crop, severe wilting and necrosis in older plants. So you can see the productivity of such crop will be very low. The quality and quantity of fiber will be adversely affected by vascular wilt. So usually whole plant wilts, but partial wilting is also noticed. In grown-up plants, wilting parts droop at tips and defoliation starts, which consequently die. Sporodochium of fungi with pinkish tinge produced on dead stem or dead portion of stem where infection is confined to one side. Discoloration of tissues could be traced to main tap root or lateral roots. So, these are the typical identification of this disease. In early stage, fungus confined to lateral roots, especially in tip region, subsequently attacks vascular bundle of the meristem, means its entry is mainly through the roots. As pathogen is facultative parasite, it survives on crop stubbles. So, if you are continuously growing sannai or sannhemp in the same field, then it is very likely that your crop can suffer from vascular wilt. So, pathogen produces pink sporodokia on which enormous micro and macro conidia produce. These conidia are actually spores of the fungus. Fungal hyphae and spores plug. Plug means just they stop the passage, plugged xylem vessels of infected part and causing the death. Now, how to control the vascular wilt? Sannhemp varieties like K12 yellow, Swin 037, Swin 053, JRJ 610 showed considerable tolerance to wilt disease. So, wherever it is likely to get wilt disease, better to grow the tolerant varieties. Wilt infestation can be avoided if crop is sown in April or early sowing can reduce the wilt infestation, and adequate drainage, clean cultivation and crop rotation should be followed. So, crop rotations are the key to reduce the incidence of this disease, and for many diseases crop rotations are useful to prevent them. Seed treatment with carbendazim at the rate of 2 to 3 gram per kg seed helps preventing the disease. Soil application of neem cake at the rate of 100 kg per hectare along with zinc sulphate 25 kg per hectare is useful. So, these treatment can reduce the incidence of this disease. Spraying of carbendazim 2 gram per liter water had been reported as effective control measure. Now, next disease is anthracnose. So, it is caused by Colatotrichum curvatum. You can remember this name. This is important disease. Anthracnose is caused by Colatotrichum curvatum. Affected seedlings show drooping. Drooping means just the kind of hanging. Infection is spread downwards and seedlings finally die. In infected older plants, it is restricted to leaves and stems, and in case of heavy infection, leaves are shaded. Spores on older leaves appear on one side of leaf, but gradually enlarge and extend to opposite side. These spores are grayish brown to dark brown and roundish or irregular. Several spores coalesce. Coalesce means they merge into each other and then they become bigger in size and cover entire leaves or large portion of leaf, may also be formed on midrib. And along with growth of seedlings, plant becomes resistant to disease. What are the control measures for anthracnose? Let us see them. And disease can be controlled through early sowing. Sowing can be done in April to mid-May to avoid the incidence of anthracnose in sannhemp. Seed treatment with carbendazim at the rate of 2 to 3 gram per kg of seed is useful, and this can reduce the incidence of anthracnose. And carbendazim, same fungicide can be used for spraying, and spraying can be done at the rate of 1 to 2 gram per liter of water. And varieties like K12 yellow and SH4 are largely tolerant to this disease. So, wherever it is likely to get anthracnose, we should choose the varieties which are tolerant to this disease. Now, next disease is leaf mosaic. So, sannhemp mosaic is a viral disease and infection is more with onset of monsoon. Whitefly, Bemisia tabeci transmits virus causing leaf mosaic in sannhemp. That means whitefly is the vector of this disease, which transmit disease from one plant to another plant. And you can see the symptoms. This is yellowing, yellowing of leaves, and then finally they can dry also. You can see the symptoms on the left side, yellowing of interveinal areas, and on the right side you can see some healthy leaves. Control measures of leaf mosaic. Early sowing in mid-April can protect crop. Sannhemp varieties K12 yellow, SH4, SUIN 053 have shown tolerance to mosaic disease. Seed treatment with imidacloprid 1 gram per kg seed. So it will help to reduce incidence of white fly, so that transmission of disease will be less, particularly at seedling stage. Bamesia tabesii can be effectively managed by spraying imidacloprid 17.8 SL at the rate of 0.4 ml per liter. See, the quantity of imidacloprid for spray is very small. So we have to be very careful in using this insecticide. The quantity is 0.4 ml per liter of water, and 500 liter of such solution may be prepared for spraying. And uprooting and removal of severely infected plant will prevent the spread of the disease. Fourth disease is powdery mildew. It is caused by several fungi. For example, Levulula torica, Odium erysiphoides, and Golovinomyces cytoserium, and it attacks only older crops, and affected plants should be uprooted and removed away from the field. So it is not that powdery mildew comes at early stage, it normally comes at a later stage of the crop. Leaf blight. It is caused by Macrophomina phaseolina. Macrophomina phaseolina. The same fungus causes variety of rots in other crop, which is jute crop. It causes stem rot, it causes certain blights, even root rot in jute is caused by this fungus. Here it is causing leaf blight. So blight starts from margin of leaf and proceeds inward. Under moist and warm conditions with intermittent rains, whole leaf may be blighted. Blighting means just it will look like burnt. But with fall in temperature along with receding rains, so disease severity reduces. Black pinhead-like fungus structure noticed on blighted site. In early morning, blighted leaves look grayish and water-soaked, which ultimately become brownish with broad yellow margin. You can see here. So these are the symptoms of leaf blight on the leaf. You can check in this picture. Subsequently, infected leaf becomes weak and droops down from plant, which gives a sticky appearance of whole field. Now, twig blight is another kind of blight caused by Conifera species, and infected plants show blighting of top portion. Plants decay from top, droop, and they can also break. Whitish to grayish mycelium with black fruiting body seen on infected parts. Spraying of carbendazim at the rate of 2 gram per liter effectively controls twig blight. So these kind of blights can be controlled by spraying carbendazim, which is also known as bavistine. It is a systemic fungicide and quite effective against many fungal diseases. Now, leaf curl virus, sannhemp leaf curl virus is observed more in crop sown during May to June, means the crop which is sown late. Early sown crop which is in April escape the infection. So by now you could understand that late sown crop or crop sown in July or say later has more problems than the crop which is sown in April or middle May. These crops actually get less of rainfall, less of humidity. Therefore, insect pest disease incidence in early crop is less. So, infected plants of leaf curl virus are stunted and leaves turn yellow, shortened and curled with interveinal chlorosis. Severely infected plants be uprooted and removed. Spray neem seed, neem based formulation 2 to 3 ml per liter. it is necessary to remove the infected plants because they can produce disease in other plants. So carefully infected plants should be removed and they should be taken away from the field. Now sannhemp rust caused by fungus Euromyces decorates, and initial symptoms of rust are small yellowish spots on leaves. As disease progresses, spores develop into raised pustules that are reddish brown in color. Severe infection, there will be premature leaf drop, reducing plants' photosynthetic capacity and overall vigor. Because of these pustules, larger area may be covered for the leaf and very less space is left for green space is left for photosynthesis. Avoid planting sannhemp in same field year after year to reduce the buildup of inoculum. That means we need to follow clean cultivation as well as crop rotation should be followed. Remove and destroy infected plant debris to reduce source of spores. Planting with appropriate spacing to ensure good air circulation to reduce humidity that favors fungal growth. Planting rust-resistant varieties of the sannhemp. Treating the seed with fungicides before planting can help reduce the initial inoculum, and regular monitoring of the crop for early signs of infection to implement control measures promptly. Fungicides can be used and follow local recommendation for fungicide use to avoid resistance development. Now sannhemp phyllodes, abnormal development of floral parts into leafy structure or vegetative structure caused by phytoplasma which inhibit phloem of plant and transmitted by insect vector, particularly whitefly. So you can remember that this disease is caused by phytoplasma which are transmitted by whitefly. Affected plants exhibit a significant reduction in normal flower formation and stunted growth and reduced vigor of plants. So you can see this picture of sannhemp phyllodes. Shoot proliferation in number 1, you can see with reduced size of leaves. Chlorotic leaves are there in this picture A, and then see picture B. This is abnormal growth, and see this picture C where normal flowers are there, and in D phytoplasma affected plant, these, and in E healthy plants in the field. Not very clear picture, but somehow you can see that the floral parts are transformed into leafy parts. Here you get clear picture. So you can see H is healthy plants. This is healthy plant and this is phyllodes plant. You can see there are no flowers. On the right side also you can see this is phyllodes plant or malformed floral parts, but they are healthy flower parts. So implementing crop rotation with non-host plant to break the life cycle of phytoplasma is a good control. Remove and destroy infected plants to reduce source of infection. Use insecticide to control whitefly by spraying imidacloprid 17.8 SL at the rate of 0.3% or thiamethoxam 25 Wg at the rate of 0.25%. So insecticides can be employed to control the vector. Now you have seen there were number of diseases which are coming to the crop of sannhemp, but similarly there are number of insect pests that can infest this crop and they can reduce the productivity, particularly fiber yield of this crop. In India there are two major insect pests of sannhemp. One is top shoot borer, Les Paracea, Pseudomonectis, and hairy caterpillar, Utethesia and Pulchella. These two insects cause severe damage every year, and flea beetle can also be there, and stem girdler is also there. These are the minor pests of this crop. Now let us start top shoot borer. This is infestation is more prominent in late sown crop, particularly when it is sown in June, July compared to sown during April and May. The main reason is that during this period, rainy period, there is more humidity which make conditions favorable for insect growth. So larvae initially bore into top part of the stem, thereby causing reduced growth and branching. Subsequently, larvae bore and feed inside the stem, causing swelling of stem, and resultant fiber becomes short, coarse, of poor quality. So therefore, this particular insect affects quantity and quality of the produce. During pod formation, larvae bore into the pods and feed the seeds. Extent of damage is maximum from September to October. You can see the damage by top shoot borer, and this is how it is entering into the pods and destroying the seed. Control measures: clean cultivation, deep summer plowing, collection and destruction of galls are effective cultural measures to control this insect. Spraying of neem seed kernel extract 5% or profenophos 50 EC 2 ml per liter or cypermethrin 10 EC at the rate of 1 ml per liter water at 15 days interval can control this insect. Some effective parasites reported against top shoot borer in India are cremester, which is Tethala, A. tetragame, and there are many more natural parasites which can control this insect. Hairy caterpillar is number 2 insect. Hairy caterpillars are gregarious, means at early or initial stage they feed on the plant in groups. Therefore, gregarious. Feed leaves completely at early stage and pods at later stage. Early sown crop, April to May, is more affected by hairy caterpillar. You can see pictures of this hairy caterpillar. You can easily identify them, and more pictures you can see. Now, collection and destruction of caterpillar at an early stage of infestation help in controlling the pest. Spraying of neem seed kernel extract or 5% or profenophos 50 EC 2 ml per liter or cypermethrin 10 EC at the rate of 1 ml per liter water at 15 days interval can control this insect. Some important parasites of hairy caterpillar are given here, for example, Phaneurus, Drino and Bracken, etc. So there are certain parasites that can be used to control this caterpillar. Now, third one is sannhemp flea beetle. Both adult and larvae are actually grubs or caterpillar, cause damage. Adult beetles make minute holes in leaves and grubs feed on the roots by mining. So you can see the leaves damaged by the flea beetle. So they have eaten the leaves partly, and you see here the typical flea beetle are doing the damage to the leaves. Next is sannhemp mirid or capsid. Minute chlorotic spots later coalesce to cause yellowing of leaves. And nymphs and adults suck the sap from tender leaves and shoots, causing yellowing of leaves. In severe attack, cause death of the plant. Stem girdler. Adult female girdles stem at 2 levels as a pre-ovipositional operation result in stem withering, drooping and finally dying above the level of the lower girdle. Dusting methyl parathion 2D at the rate of 25 to 30 kg per hectare keeps the pest under control. Number 6 is white fly. Besides spreading some diseases, viral diseases to other, it also directly damages the crop, and nymph and adult suck the sap from the underside of these leaves. Severe infestation result in premature defoliation and development of sooty mold, because these insects can release honeydew which attracts certain fungus or which gives black color. Spray imidacloprid 17.8 SL at the rate of 0.3% or thiamethoxam 25 Wg 0.25%, and on an average 500 to 600 solution can be made and sprayed. Spiny pod borer larva feeds on growing tips blossom and developing pods. Damaged parts will drop down due to feeding. Seeds are either partially or entirely eaten, and considerable granular frass and silk are present. Externally, pods have punctures. You can see the damage being done by the spiny pod borer. It is damaging the pod of the plant. Now, let us go for retting and extraction of fiber. If you have grown this crop for fiber purpose, then this is an important activity. So, first you need to go for retting, followed by extraction of fiber. So, fiber in bark is separated from woody portion by action of microorganism present in water. It involves steeping and keeping stems submerged in water for certain periods. You need to do this process under anaerobic conditions. Retting time depends on water temperature, locality, time of year, weather conditions, depth and source of water, thickness of stalks, and quantity of straw in relation to volume of water. So, retting time of jute fiber is also important, and normally retting time needed by sannhemp is 3 to 5 days in September, while 7 to 9 days required to complete retting process in December when water is cold. So, naturally the microbial activity which is responsible for retting process is affected by temperature. If temperature are too low, which is the case in the month of December, then it will take longer time. If it is being done in September, when temperatures are higher compared to December, then the time for retting will be less, which is normally 3 to 5 days. So, because the cold water in December will result in lower microbial activity compared to warmer water in September, which encourages microbial activity. In most cases, retting tanks are shallow, muddy ditches adjoining the fields where runoff water from a shorter spell of monsoon is collected. In villages, there may be some ditches, some ponds, and some water storing structures where retting process can be made or can be done. And also some people like to make the cemented tanks, and they are also good for retting purposes. Here the loss of water will be less because the bottom you can cover with polythene or some cemented material. So, water storage is good under cemented tanks. But earth pits, drains, streams and backwater pools of river are also used. So, in certain cases these streams can be used, but condition is that the flow of water should be less. If flow of water is very fast, then decomposition process may take longer time. If more than one rat is to be carried out in a pool, sufficient flow of water must be ensured to avoid fouling the water which discolors the fiber. So, it is quite simple that overcrowding of the bundles should be avoided, means in lesser place or lesser water you cannot put too much of bundles or too much of biomass. It should be in proportion to the amount of water which is available. So, shallow water of 1 meter to 1.5 meter depth can serve the purpose. So, you need not to go for deeper placement of the bundles for retting. That may involve extra energy and extra labor. So, therefore, the best way is to keep bundles at 1 to 1.5 meter deep in the water. So, during steeping, bundles arrange side by side, preferably in a single layer in water. Try to avoid making the stacks of bundles. Single layer is better. Bundles are weighted down with some cement blocks to a depth of 10 to 15 centimeter in water. You will require some weight so that bundles can be merged into the water. So, weighted material which release tannins such as banana logs should not be used because the quality of the fiber will be affected. The coloring of the fiber can happen because of such materials. So, better to use cemented logs or some heavy cemented things. If on testing the retted stalks fiber is found loosening, it is considered ready for extraction. So farmers have idea when the retting is complete, means then fiber can be easily removed from the stem of sannhemp. Now this picture you can see is steeping of sannhemp. So here you can put it into the water and then cover. On the right side you are seeing extraction of sannhemp fiber. Softening of fiber takes place due to action of enzyme released by bacteria. As many of you know that this particular process goes under anaerobic conditions. So here most of the organism, including fungi, bacteria, they are anaerobic in nature and they decompose certain constituent, thereby making the separation of fiber easy. So precaution must be taken against under-retting or over-retting of fiber. In both cases, whether it is under-retting, retting or over-retting, in both cases the quality of fiber is compromised or quality of fiber is reduced. In under-retted condition, removal of fiber will be difficult as bark adhere to extracted fiber. So here the separation may be difficult, and at many times there will be breakage of breaking of the fiber if it is under-retted, and it will also consume more time, more labor. What happens when it is over-retted? Over-retted condition, microbes break down tissues surrounding fiber and causes weak fiber. So in both cases the quality of the fiber is reduced. Extraction of fibers of sannhemp is more cumbersome than jute here. Beat and break and jerk, this is the common manual method. Beat, break and jerk method unsuitable, as there is tendency of fiber to stick to wood is more and fiber gets entangled with broken twigs. So this technique has some limitations also. You will take 4 or 5 stems together in hand and beat them and give jerk and then try to separate fiber, which can result in broken fibers. When retting is completed, bundles dashed against water to 3 to 4 times to remove excess lignin, and then bundles moved in water to and fro. So you can clean it. Vast bundles are stacked vertically for dripping of water from sticks and fibers, means you need to take out water from the fiber. Fiber can easily be extracted when vast bundles of sannhemp are moist. Fiber from each plant is extracted or peeled off manually from bottom to top side in a long strip. If retting is satisfactory, fiber peels off smoothly from inner stem, but there may be difficulty if retting is not complete or over completed. Extracted fiber dried in sun and put up in twist and bundles for marketing. So drying in sun is necessary, otherwise this continuous moisture in fiber can deteriorate the quality of the fiber. It can change the color, it can decompose the fiber or can weaken the fiber. So therefore drying in sun is necessary, and then one can make the twist and bundles and keep it in your home, or it can be sold in the market. Now we go to the fiber quality. So definitely fiber quality is very, very important, because the fiber, if it is poor in quality, will fetch less price in the market. If it has good quality, better quality, it will fetch more price. So it is necessary to understand what are the quality parameters and what do we mean by fiber quality. So sannhemp fiber is lignocellulosic in nature, and about 3 to 4% of total green biomass is your sannhemp fiber. Major chemical constituents are alpha cellulose 78.3%, hemicellulose 3.6% and lignin 4%. So exact composition of fiber can vary depending on factors like plant genetics, growth conditions and processing methods. So definitely number of variables or factors can affect the yield of the fiber, particularly genetics. You have already seen that some of the varieties give more fiber yield and some give less fiber yield. Similarly, quality. Some varieties will give you more quality, better quality than other variety. So similarly, environmental conditions, climatic conditions and soil health also affect the fiber quality. And of course, the processing method. Everything is fine, the yield is good, quality is good, but processing is bad. So bad processing can result in bad fiber quality. Therefore, we need to have the best techniques and methods and timing of processing. Like other natural fibers, sannhemp fiber is biodegradable, renewable and environmentally friendly, making it a sustainable alternative to synthetic fibers. So people try to use these synthetic fiber more these days because they are cheaper, but they have several or negative consequences on the environment. But these kind of fiber, they are natural, they are biodegradable, therefore they are environment friendly. As seen in last slide, the important constituent of fiber are cellulose, hemicellulose, lignin and pectin. Now cellulose is a complex carbohydrate that provides strength and rigidity to plant cell walls. Cellulose is main component of most plant fibers and contributes to their mechanical properties. So it is the main organic compound which is responsible for mechanical strength of the fiber also. Hemicellulose is another organic compound, group of polysaccharides that provide additional structural support to the plant cell walls. So cellulose, hemicellulose and pectin are the important, lignin are the important constituents. So lignin is a polymer that is binding material in plant cell walls, providing rigidity and resistance to microbial degradation, affects flexibility and durability of the fiber. So lignin is also important constituent of fiber quality like cellulose or hemicellulose. Then there are some other components like pectin and minor components. So contains a small amount of pectin, proteins, waxes that contribute to their overall properties. Now fiber quality. Important properties of sannhemp fiber that determine its quality are strength of the fiber, fineness of the fiber, length of the fiber, particularly ultimate cell, and color of the fiber. So these are the four important fiber quality parameters in sannhemp. So let us start with strength, usually expressed as a tenacity. Tenacity, it is a ratio of the breaking load which is expressed in gram of fiber to the fineness, and fineness of fiber is expressed in respect of tex. Tex is the unit TEX, which is most important properties that influences yarn quality. So yarn is your kind of thread that is prepared from the sannhemp fiber, yarn. Now the second property is length. The breadth, length is to breadth ratio of sannhemp fiber is moderate amongst vast fiber crops, 450, higher than jute, mesta and sisal, but lower than rami and flax. All these are fiber crops like mesta, jute, sisal, rami and flax. High amount of alpha cellulose as well as its degree of polymerization or chain length makes this fiber very much suitable for production of high quality paper. So many a time the fiber of this crop, sannhemp, is being used for high quality paper making. Properties of fiber are mentioned in next slide. Now you can see properties of sannhemp fiber, properties and measured value on Y column. So, first you see the first column, properties are given. First of all, you see ultimate cell. Ultimate cell is number 1. You go for length. Length is given in millimeter. So, measured value ranges from 5 to 20 millimeter. Now, see the breadth is 10 to the power minus 3 millimeter, or it can be simply written as 1 upon 1000 millimeter, or you can write in millimeter also 0.001 millimeter. It is like this. So, you can convert this 10 to the power minus 3 millimeter into 0.001 millimeter. So, this is the way some mathematician write it, and it varies from 12 to 35, the measured value. Length breadth ratio is 450, and this kind of properties in sannhemp fiber has been reported by Chaudhary and co-workers 1997, Day and co-workers 2006, and Kundu and co-workers 2005. Now, you can see more properties of sannhemp fiber, and number 2 is your filament. On the right side, you can see the measured values. So, filament, you can have gravimetric fineness. It is expressed in tex. TEX tex is the unit. The measured value may range from 2.5 to 3.5. Then comes the tenacity. It is gram per tex. It is varying from 30 to 40. Then extension at break, percentage 2.5 to 3.5. Modulus of torsional rigidity 10 to the power 10 dynes per centimeter square. It varies from 1 to 2. Then flexural rigidity dynes per centimeter square 125 to 175. And then transverse swelling in water. It is being expressed in percentage 18 to 20. And again it has been reported by Chaudhary co-workers 1997, Day et al. 2006, and Kundu et al. 2005. Et al. Means co-workers and associates. You can see some more properties. The third important property is related to bundle, and in second column you see they are the measured value. So, tenacity gram per tex is 15 to 35. Then your true density gram per cubic centimeter, it is in gram per cubic centimeter 1.53 is the true density, and then apparent density is gram per cubic centimeter, it is 1.34, and moisture regains in percentage at 65% relative humidity, its value is 10.5. Moisture regains percentage at 100% relative humidity, it is 28.5, and degree of crystallinity percentage 57.7. So, these are different quality parameters of this sannhemp fiber. So, dear students, hope you enjoyed this lecture very much. So, thanks a lot.